Okay. All through these days, throughout the conference, we have had amazing sessions where we realized that data curation, before that, data generation, procuring data, and maintaining that data are fundamental to any biological experiment or any experiment we do in our labs. And equally fundamental is curation and maintenance of that data. So this is a quote from John Naisbitt, but I got to hear it from my professor, that we are drowning in information but starving of knowledge. And definitely, I started this work with this belief. So the work I'm presenting here today is mining drug target mutations to assess drug efficacy. And please don't go with the title so much. As I walk through my presentation, you'll realize the steps we traveled for this work. And I'm from University of Delhi, New Delhi, India. So this is the work I'm presenting here today is a part of the objective where we were trying to study the mechanism or the fundamental factors affecting antifungal resistance, or factors underlying resistance in fungal pathogens emerging during COVID, and because fungi has recently taken its place as a pathogen. Earlier, the era of resistance has been dominated by bacteria mostly, and fungi as a pathogen is being taken seriously as of now. So in this study, one of our objectives was to decipher the linear relationship between fungal drug resistance and the mutations in their target sequences and how they affect the resistance pattern. But as we walked through our objective, what we ended up, at present, what we have and what I'm presenting in the talk is, we developed a generalized natural language processing pipeline, text mining based pipeline, where from systematic extraction of data, we moved on to visualizing that data by implementing an R-based application. So this is the work I'm presenting here today, a natural language processing based pipeline, and then it's the visualization of data extracted through that pipeline in an R-based visualization Shiny application. The name of the application is Mute Drug Spot. It is freely available. So as I walk through the presentation, I would like a minute of your time on the background with which we started this work, then the methodology, and the case study, which will be the fungal drug resistance here, and finally acknowledgements. So as I just mentioned, with the advent of COVID-19, all of us were scared and were at homes. Equally developing with COVID-19 were other pathogens which were not being taken, I would say, seriously by human race as of now, but fungus as a pathogen has been enveloping globe. And as you can see in the red areas marked here, that these are the areas where fungi has already developed as a serious pathogen, majorly in hospital-acquired infections. It started with a case in 2009 in Japan and South Korea, and gradually it's moving all over the Asia-Pacific and the globe. So as we could see that in hospital-acquired infections caused by fungi, as the fungi enters the patients who are already suffering from major diseases like they are admitted in the hospital with diseases like cancer or other fatal diseases, but they end up dying due to the fungal infection. So this is the case with hospital-acquired fungal infections. Then while investigating this, our first step obviously was data collection, But as we moved on to data collection, we could realize that with the advent of sequencing, yes, we had lots of sequencing data available publicly on databases like NCBI PubMed and everywhere, but the arrangement and the organization and the coherence of that data was difficult for us to derive any meaningful conclusions. So we started with streamlining the search word first. For that, we used medical subject headings, the mesh library. We also used some publicly available utilities, that is the NCBI e-utilities. There's a bio-entree package which helps us use the e-utilities of NCBI to extract exactly what we want from the papers available. And then through BioPython and other packages, we could streamline the search word, and we moved on to extraction of the data which we exactly wanted. And here, as you know, as I've been discussing as of now, that the data we were focusing at was drug and protein. Protein is the drug target. So the drug, its target, and the mutation which makes that pathogen resistant against that particular drug. So this was the data we were targeting to extract from the articles we had filtered through the search word. Then, after the extraction of data through this BioPython packages and the mining pipeline, what we developed is we moved on to parsing that data using the natural language toolkit. It is a freely available package. All the everybody who's working in NLP and machine learning might be aware of this, that this is the favorite or first choice of people who work in NLP. So here we pass the data extracted from these papers into text, sentences, and words, and text, sentences, and words targeted at our objective data, that is drug, the drug target, and the mutation. So while we were filtering our data, the major problem we faced was the collection of that data. These are the different resources we used to streamline, filter, and curate our fungal data. So these are publicly available databases. All of us know NCBI, UniProt, but some of these, EMBL, EBI, but some of these databases like GAFFI, that is Gafi, Global Action Fund for Fungal Infections. These are NGOs working for fungal infections and collecting data like this. This is FRAC, Fungal Resistance Action Committee. So all these bodies have publicly available repositories where we could extract clinically relevant data of fungal pathogens. So these were the sources through which we tried to streamline our data. And yes, IMA, International Mycological Association. Then after streamlining our data, our next thing was to put it into the format which we could extrapolate on our visualization application. So for that, we had, using the resources which I showed just now, we constructed 3 libraries of one of the drug, one of the pathogen names, and one of the gene or the protein. The gene or the protein here is the drug target. So using these 3 libraries and the data extracted as shown in the previous slide, we could come up with the format of our data into this form. And finally, then we implemented it into a Shiny-based application. This application is freely available on our GitHub link. And it is very simple to use, as we discussed in some of our, as we heard in some of our previous talks, that Shiny applications are really easy to use, where you just have the R environment in your system, and you can log into this Shiny package. And using that, the user just has to input his or her data in the form of the list of mutations, the name of the gene, and on the name of the drugs which you want to plot. So using the application that I just showed, we get plots like this. I'll just take a moment to explain this plot. So we call it mute plots or mutation hotspots. So on the x-axis, you have the length of the protein. On the y-axis, there's no significance of the length of these lollipop kind things. But on the y-axis, you have the exact locus where the mutation is caused. Here, as you can see, Y is replaced by C, that is tyrosine B16 at the 33rd position. And this mutation in this protein will make the entire pathogen, or will make this pathogen resistant against this drug, which is shown by the black color here. The color black means fluconazole, itraconazole, voriconazole. These are 3 antifungal drugs commonly used against this pathogen. So these are 3D plots which we ended up generating using our application. And besides fungal or pathogens or any pathogen-based diseases, these are also of relevance where we want to describe or show basically these are 3-dimensional plots. So we can picturize anywhere where we have 3 dimensions, we can make use of these plots. So as I just mentioned, the pipeline is now a generalized pipeline, but we started it for our fungal work and ended up making the pipeline. And initially we started with 20K articles, which were resistance specific, but fungal resistance specific around 13.5K research articles. We were able to filter through our NLTK pipeline 6.7K articles. And finally, through these, the parsing, we could extract around 3688. The number is a little more at present. Unique mutation drug relations. So mutation drug relations as in the drug target, and the mutation in it. And these are the other data points: the host, the pathogen, the genes, and the drugs and the diseases. All this data we have put and implemented into a database which is publicly available, and this is published, this has recently been published. So we have ended up implementing and publishing the data first in the form of a database, and we are in process of publishing this pipeline as a whole. Right now, I'll move back to the slides a little bit. Yeah. Right now, these pipelines, as I'm showing, are in a 3-step process, but we are in process of compiling it together in platforms like KNIME, where the user can come directly with the search words he or she has, and finally end up with having the plots of this kind or other visualization tools which we have manufactured in this pipeline, implemented in this pipeline. So these are the other data points. We could do analysis like this by combining these both pipelines, the text mining based and the R-based visualization pipeline, and implemented it on our database. So that's it with my study. So I would definitely want to push in this key takeaways. The data visualization definitely lies at the root of any data analysis pipeline, but loss or misinterpretation of data is an essential consideration while generating the data points we do and the filtering parameters we put into the coding pipeline. And yes, the route of our methodology towards the objective of our work may not always be straightforward. As long as we are sure about the objective, the rest goes with necessity is the mother of invention. So I would like a moment on the objective we had passed in 2019 when we started this work during COVID. So There is also sound, but that's okay. Please.
They came, they conquered.
It was that they came, they conquered, and they are here to stay. And the stakes are high because drug industry is booming, but definitely criticality of life is higher.
Time to act is now.
There are different NGOs working on this. Time to act is now because a little late will be fatal. This is a patient who was admitted for different reasons but ended up dying due to fungal infection. So finally, I thank all of you first for bearing with me during the presentation. I thank my supervisor, Dr. Manish Kumar, Dr. Neelja Singhal, Sultan Sayed, and Ayush. These are the other authors in this work. And I thank my lab members who have become a family. And also I thank my family members and all our family members because they bear with us when we are in our labs or before the computers. And also I thank my university institution and also my PhD stipend for funding me. Thank you and have a great time in Singapore. Thanks a lot.
Okay, let's take questions. Kita?
Hi, Aakriti. Nice talk and congratulations on your paper.
Thank you.
So, yeah, I have seen that you have used the different drug-protein interactions through the literature. So sometimes what happens is that they're like normal computational docking analysis also that checks the binding between drugs and proteins. So which doesn't have that much of experimental evidence, you can say. So have you taken any additional measure to filter those interactions, or have you included them too?
Thank you for your question. Since our input data was specifically targeted on the genomic and proteomic input, therefore we just worked on the sequential data, which was taken from the public literature, which was already available. We did not have to, or we did not rely on other efficacy or binding parameters. As if a researcher got sequenced a mutation, we treated that as a mutation and making the drug ineffective.
So in future, are you are you planning to consider those things in future development? Because I have seen hospitals like AIIMS and all, also they have a lot of fungal infection cases, so yeah.
Yes, thank you very much for your suggestion. Yeah. But I would like to add on to this that since now this is a pipeline where the primary target is of taking the text and making it easily comprehensible. So I think the initial filtering parameters, we have already filtered it to a level where we can execute it to the next level. So for the it'll go to another dimension if we also include the structural part in it. But yeah, and also I don't have an expertise in structural mathematics. So
nice talking to you. Thanks.
Thank you so much.
Okay, you can have the next question.
Yeah, just a bit of a related question, I think. I mean, it's a very nice project, I think, in an area that is needs a lot more attention. And maybe you said this. Have you thought of expanding this to cover Have you thought of expanding this to cover a wider range of fungal biochemistry and physiology? Because I think there's a lot of issues that remain unresolved. This idea of the thermotolerance hypothesis that there are going to be more fungi that will become human pathogens as we move deeper into the Anthropocene because of increasing temperatures. And so I think there's a lot of really interesting potentials that you have here.
Thank you very much for your comments. Yes, about the expansion of this project to specifically to fungal pathogens. About this project is, I think, not our objective at the moment, because this pipeline we feel is complete in itself and a generalized one which can be used for in precision medicine, cancer, and other anywhere where we have 3 dimensions of the data to visualize. So as far as our fungal project is concerned, we are moving towards the co-occurring mutations and mutation patterns in high priority fungal pathogens. There's a WHO list of critical pathogens, which has recently been released. So we are working on those to assess different parameters of the mutations which could make them resistant against drugs. So that is in our pipeline as far as my PhD work is concerned.
Very interesting, thank you.
Thank you so much.
Yep, thank you for the questions. I may just have one actually. So would this be sort of like an ongoing platform where you incorporate more information because there might be more literature coming out and you already have the platform? So is that going to be a resource for the community, for example?
Thank you so much for this question. Yes, that is in the, in our heads. Basically, so the application that has been released, it is, we are using it ourselves but we haven't been able to, as of now, link both the text mining and the visualization thing. We are in plans of doing that, and yes, starting with the collection of data at the first, that we just type a keyword and the data is extracted, filtered, and finally moved on to this application. We are in a process of that.
Very great. Thank you.
Thank you so much. Thank you, everyone.
Okay, let's thank our speakers.